Here we are back in Chapter 5. We're halfway through Chapter 5, which is all about the structure and function of large biological molecules. We know that there's four major categories of these large biological molecules. We've already talked about the carbohydrates, which would include your sugars and starches and the like. And then we talked about fats, which are not normally considered polymers or macromolecules, but they are large biological molecules. And now we're going to move on to the last two. The last two are going to be proteins and nucleic acids. And right now we're going to go ahead and hit proteins, which is probably the biggest one of Chapter 5. And so we're going to take it slow, and then we'll stop, and then we'll do a different video on nucleic acids. So just as a quick overview of the chapter, all living things are made up of these four classes of large biological molecules, and we call these large molecules macromolecules. And again, here's one of our main ideas, that structure and function are inseparable. And that's going to become so important right now when we talk about proteins, because proteins are so big and so complex, and they bend and fold and twist and shape themselves into three-dimensional shapes, and that three-dimensional shape is just as important as the chemical formula that makes them up. So let's go on to proteins. Concept 5.4 says, proteins include a diversity of structures resulting in a wide range of functions. Proteins are the building blocks for you and for all living creatures. They're so important that their very name comes from the Greek word proteos, which means first or primary. Proteins make up more than 50% of the dry mass of most cells. So if we were to take your body and dry you out like a piece of beef jerky, and get rid of the majority of the water, which is a huge portion of your body, well, what's left, the dry portion, about 50% of that, or more than 50%, is protein. And proteins have a huge variety of functions within living creatures. Some of them are structural support, some of them are for storage, transport of materials, communication, movement, defense against foreign substances, things of that nature. There's a huge number of jobs. And these proteins are so large and so complex that they form unique individual three-dimensional shapes because they twist and fold and bend onto themselves, and we're going to talk about them. They are the most sophisticated types of molecules known, the most complex types of molecules known. So let's take a quick look at an overview, and then we'll kind of look at each one of these sort of step by step. And the good news is that the textbook and the notes kind of gives a short little video clip or animation for each one of these. So just as a quick overview, there are some proteins that are enzymes. We've already talked about this word enzymes, so enzymatic proteins, we'll discuss them. There are some proteins that are used for storage, so storage of energy in a way. We'll talk about that. Egg whites is a good example of storage proteins. Some of them are defensive. In other words, the proteins that we call antibodies that help keep our bodies safe from viruses and bacteria and the like. Some of them are transport proteins. They help move materials in and out of the cell and throughout the body. Some proteins are hormonal. In other words, they are messengers that help coordinate the functions of the body. Some of them are involved in movement. In your muscles, you have proteins called actin and myosin, and they work together like a ratchet strap to shorten or lengthen the muscle fibers, which allows you to move your body. Some of these proteins are receptor proteins, which allow messages to be sent between between cells, or say between parts of your nervous system. And then other proteins are structural proteins. They are the building blocks for your body. So the things that make up bones and ligaments and connective tissue are structural proteins. So we're going to take a quick look at each one of these and just hit the highlights, and then there's a short little video animation that goes along with each one as well. The first category is enzymatic proteins. Enzymes are what we call catalysts, and catalysts are something that speed up a chemical reaction without being used up or consumed themselves. So here in this little diagram you have a chemical reaction that's going to take place where we're breaking apart these two pieces, and this enzyme is kind of like the tool that gets that done, or the tool that speeds up that process. So a lot of proteins are enzymes, and enzymes are chemical catalysts. In other words, they're a tool that speeds up a chemical process without being used up itself. So if you have like a shop at home, or you have a garage where you work on your car, you're going to have all sorts of tools. Say, for example, you're taking a bolt off of your car and you're using a wrench, a box-end wrench of some kind. Well, that wrench is a tool. It's kind of like the catalyst that gets the job done faster. Perhaps you could do the job without the tool, but it sure makes it faster and easier to use the tool. And at the end of the job, the tool has not changed. You can reuse that tool over and over again, and it doesn't change the tool itself. That's pretty much what an enzyme is. So just a little background information. Enzymes are a type of protein that act as a catalyst to speed up chemical reactions. So again, think of it like a wrench or some other kind of tool that helps get the job done faster and easier, but the tool itself is not changed. There's a quick little animation video here about enzymes, so if you get the chance, right-click and hit play on that. The next type of protein we're going to look at is storage proteins. So this is for the storage of amino acids, and this is, for example, in seeds or in an egg, which is basically just an animal seed. You have storage proteins that are there. Within that chicken egg, that baby chick has no way of getting food from the outside world, so there's a huge amount of storage inside that chicken egg in the form of this is so hard to pronounce. Ovalbumin. Ovalbumin or ovalbumin, depending on who you ask. These are amino acids that the chick needs to build itself, and they're stored in this protein inside the egg along with the chicken. And again, there's a short little video here about storage proteins, how they can be broken apart and release amino acids. And we're going to find out what amino acids are. They are the building blocks for other proteins. So these proteins are kind of like storehouses for the building blocks of other proteins. You can right-click on that and hit play. Some proteins are basically our acting army, defensive proteins, antibodies, and their job is to search out and destroy, say, invading viruses or bacteria, and they protect us against disease. So these antibodies is what we call them. They can attach to certain sites on the virus or the bacterium and either disable it or destroy it. So some of these proteins are all about our immune system. Again, there's a short little animation. Right-click and hit play. Transport proteins are all about moving materials around. A good example of a transport protein is hemoglobin. That's the iron-containing protein in your blood. It's also the material with the iron that gives blood its color, and it allows the transportation of oxygen throughout your entire body. So there's lots of different proteins that move materials throughout the body or into the cell and out of the cell. And those would be called transport proteins. Again, short little video there. Right-click and hit play. Another type of protein is hormonal proteins. Hormones are basically chemical messengers, and they coordinate body systems and how body systems work together. A good example of a hormonal protein is insulin. Insulin is a hormone that's created by the pancreas, and it helps regulate the amount of sugar in your blood. The amount of sugar in your blood can't go too high or too low, and so insulin is a chemical messenger, a hormonal protein that helps keep this balance and keep this homeostasis, which is a word I think we've already mentioned, but this balance between high and low to where your body isn't having problems because the amount is either too great or too little. Another short video, you know what to do. Here we have some proteins that are involved in movement and motion. They're called contractile or motor proteins. And these proteins actually help move your body around. And they are the ones that, for example, if you have small creatures like unicellular creatures or other small creatures that have cilia or flagella, which is like the little tail that you'll see on some microscopic creatures, these are the proteins that are responsible for that motion, that movement. In larger creatures like, say, an elephant or a cow or a human, inside the muscle cells you have actin and myosin, and they work together like with a ratcheting mechanism to allow your body to move. Again, a short little animation all about the contractile proteins that allow for movement. And then we have receptor proteins. Receptor proteins allow the cell to respond to some sort of stimuli, to some change in the environment. So receptors are built into the outside portion of the cell, and receptor proteins allow for certain molecules that carry a message to go from one cell to the other. And this is very important, like in the nervous system. The nervous system uses a combination of electrical impulses and chemicals to send messages back and forth. So if you reach out and touch a hot stove, that pain signal travels via electricity and chemical messengers from one nerve cell to another, all the way up into your brain, and then your brain sends a response back. So these receptor proteins are kind of like the locations where these chemical messengers that jump from one nerve cell to the other get sent across that gap called the synapse. Of course, a short little animation for the receptor proteins. And then I think the last one here is structural proteins. Structural proteins are kind of like the bricks and mortar that hold your body together. A good example is keratin. Your fingernails and your hair and the horns of a cow and the hooves of a goat, feathers of a bald eagle, they're all made up of keratin. And keratin is a tough material made out of strands of protein. The silk fibers, or the types of fibers that are used to make spider webs that the silkworms wrap themselves in to make a cocoon. Those are all types of structural proteins. Collagen, which is found inside your body, collagen and elastin in your skin and in your connective tissues, like your ligaments and tendons. These are all structural proteins that help hold your body together. Of course, there's a short animation for that as well. So I told you that proteins had a lot of jobs, and they do. I mean, we have proteins that are enzymes, we have proteins for body defense, we have proteins for storage, we have proteins for transport, hormones, which are chemical messengers, receptor proteins, motor-related, motion-related, contractile proteins, and then structural proteins. So lots of different functions, and they can have so many functions because there's so many kinds, or such a wide variety of proteins. So let's kind of drill down and look at what makes a protein and what makes it unique from some of the other macromolecules. Now the cool thing about proteins is there's such a huge variety and they're so large and complex, but no matter what they do, all proteins are somewhat similar in that they are unbranched polymers. In other words, they're long single chains constructed from the same set of 20 amino acids. And these long chains are called polypeptides. There's that word again, poly. So we're going to find out a peptide is basically the link, and a polypeptide is what you call the entire chain of these amino acids. So, would you say that a polypeptide is the same as a protein? Are those two words exactly the same? Well, some people sometimes use them to mean the same, but actually they're not. The polypeptide is the long chain of amino acids, but a protein, a biologically functional protein, is actually that chain folded and coiled and shaped into a specific three-dimensional shape that has a particular job. And that protein, which is the folded and turned and twisted and properly shaped polypeptide chain, can be made of a single chain, or it can actually be made of more than one chain. But the protein is that chain folded into the right shape where that it actually works and does its job. So peptides are the short chains of amino acids. Those are like the links. They are linked together by something called peptide bonds, and that forms these polypeptide chains. And then when those polypeptide chains are bent and formed into their proper shape, we would call them a protein. And that 3D shape matters a lot. As a matter of fact, that's what makes the difference between a working protein and a non-working protein. So think back to some of the other things we've talked about. So think about like that word polysaccharide. These were sugar monomers. Those were like the links connected into long chains by glycosidic bonds. Well, here we have polypeptides. Instead of polysaccharide, it's polypeptide. Peptide tells you we're dealing with the amino acids here in proteins. And so polypeptides are short chains of amino acids linked by peptide bonds. So you're going to kind of see this repeating pattern to where it doesn't matter if we're talking about proteins or carbohydrates or nucleic acids, we have chains made up of smaller pieces. So if we're talking about these proteins, or these polypeptides that can form into proteins, what are the links of these chains? Well, the links in a polypeptide chain are amino acids. And amino acids are organic molecules with a carboxyl and an amino group. And we think back to our functional groups. Here's a carboxyl. It's got the carbon with a double bond to an oxygen and a single bond to an OH. And then the amino group contains nitrogen. Here's the first time where nitrogen is part of the mix. So if we were talking about carbohydrates and some of those other big molecules, there was really no nitrogen involved. But here, when we're talking about these amino acids, we have amino groups, which involves a nitrogen right there connected to two hydrogens. And just like we've seen with some of the other large molecules, the main part of these amino acid molecules are all the same. They have this carbon attached to a carboxyl group and an amino group, but where they differ is in what's called a side chain. This side chain, sometimes called an R group. And here R is just like X. It's just a stand-in for whatever that is. So let's take a quick look at a picture. So here's an amino acid. We have the carbon right here. It's attached to an amino group and a carboxyl group, and then it's got a hydrogen attached here. And the difference between all these 20 amino acids that we're going to take a close look at is the R chain. And again, R is just a variable. It could be X or Y or any variable that you're using in math class. R is the variable that represents whatever this side chain is. And there's going to be different side chains depending on what amino acid you are. Now there's 20 amino acids, and the good news is we don't have to memorize those, but I do want to kind of show you a quick overview of them. So if you look closely, you'll notice that they're all very similar, but this R chain, this variable chain, is what makes them different. And there are 20 different ones. Some of them have nonpolar side chains, and if it's nonpolar, that means it's hydrophobic. Some of them have polar side chains, which means they're hydrophilic or attracted to water. And then some of them have different charges and would be considered either acidic or basic, depending on their charge. But altogether, there are 20 of these amino acids that are linked together to create polypeptides, which can then fold themselves into certain shapes that we would call proteins. Just as a quick repeat, if you look on page 79 in your textbook, whenever we get textbooks or if you have textbooks, there's a great chart in there on page 79. But these are amino acid polymers. Now we've already talked about some of the other types of polymers when we talked about carbohydrates and that type of thing. So these amino acids are linked by peptide bonds, just like in the carbohydrates you had monosaccharides that were linked by glycosidic bonds. And a polypeptide is a polymer, long chain of amino acids, just like a polysaccharide was a long chain of monomers, sugar monomers, glucose usually. So a polypeptide is just a long chain, a polymer of amino acids. And remember, we have 20 amino acids to choose from. And these polypeptide chains can be just a few amino acids long, or they can be made up of thousands of amino acid monomers. And each polypeptide chain has a unique linear sequence of amino acids. And remember, we have 20 to choose from. So you can have lots of different combinations along this long chain. So imagine creating a chain where you have 20 different colors of link, and you have a length of chain that could range in size from just a few links to thousands of links. And within those chains, they could be any variation of the different colors of link. So you can create a huge number of these polypeptide chains. Now each one of these chains is going to have a carboxyl end and an amino end at the say the left and the right end, if you want to think about it that way. But in between, you have this huge possible variety. Just like with some of the other chains that we've already talked about, it's a dehydration reaction that occurs to link these chains and make them longer and longer. So take a look here. We have 1, 2, 3. These are 3 different amino acids. Notice the little backbone part is all the same, but the R group, the side chain, is different. So these are actually 3 different amino acids. These two are already linked together. That linkage is called a peptide bond. And here we have a new peptide bond forming. It's a dehydration reaction. And now, instead of 2 chains long, we have a 3-chain polymer, a 3-chain polypeptide. So notice that the far ends, the left end and the right end, they're going to end the same way. It doesn't matter if this chain was 3 amino acids long or 300 amino acids long. It's going to have an amino end, is what's called the N-terminus, the end point, and a carboxyl end, the C-terminus. But in between, you could have any number of variations between those 20 amino acid possibilities, plus how long the chain was, how many links. So every specific polypeptide, every chain has a particular sequence of what type of amino acid, which is determined by its side chains, and then how many, and in what particular order. Because you could take this 3 amino acid chain and you could swap the order around and it would be a different polypeptide. It would create a different protein in the end. These polypeptide chains, they start out as long linear chains, but that's not the way they stay. Because we're going to find out some of the different chemical bonding that goes on, they tend to twist and form themselves into three-dimensional shapes. So for a polypeptide to be a protein, it has to be in the proper shape. And it's going to consist of either one polypeptide or sometimes more polypeptides twisted and folded and coiled into a particular shape that has a particular purpose. Here you have what's called a ribbon diagram or a ribbon model on the left, and here you have like a 3D space filling model on the right of a protein. It's made of a polypeptide chain, but it's twisted and folded in a certain way to create what we call a functional protein. And the sequence of the amino acids, what are the links in that chain and how many links are there, that is ultimately going to determine the three-dimensional shape of that protein. So a protein structure determines its function, and you've heard that over and over again. Think about it like a lock and a key. So if you have a particular site, like this lock site, well, there's only one key that fits that lock site. And in biology, there's lots of sites, say on the membrane of a cell or some other part, where it's shaped in a certain way, and only a certain molecule that's shaped that way will fit that particular opening and perform that particular job. Here's a real-world example of a model of a protein from a flu virus. On the right is the bended and folded and shaped protein from a flu virus, and this is the antibody protein. And notice, remember, antibody protein is like for protection. It would be trying to locate this virus and disable it, or you could say kill it in a way, although it's debatable whether viruses are alive, so let's say disable it. Notice how well they fit together, almost like a lock and a key. If they don't have the proper shape, it won't fit together in the right way, and this antibody protein will not be able to do its job. So the structure and function of that protein are related together. So a polypeptide is not the same as a protein. A polypeptide is the long chain of amino acids, but until it's folded and shaped in the proper way, you wouldn't call it a functional protein. And you can compare that to like a length of yarn. Ooh, I misspelled it. That should be yarn. Let's fix that. A length of yarn is not the same as a sweater. The sweater is the length of yarn, but it's shaped and folded in such a way to create this functional object. That's exactly the same with the protein. The functional protein is going to be one or more of these long polypeptide chains, but twisted and folded into a particular shape for its particular job. Most of the time, these shapes tend to be what we would call globular, kind of like spherical in a way, but sometimes they're more kind of straight, fibrous, long fiber shapes. But they're going to take a particular shape, and that's going to affect its particular job. Now, because proteins can be so complex, we talk about their structure in 4 levels, and we call those primary, secondary, tertiary, and quaternary, or quaternary. So these are the 4 levels of protein structure. We're going to find out that all proteins have at least these first 3 levels, primary, secondary, and tertiary. But the 4th level we only see when the protein is actually made of 2 or more polypeptide chains. So if the protein is made of only one polypeptide chain, you're going to see the first 3 levels of structure. But if the protein is made of more than one polypeptide chain, you'll end up with all four of these levels of structure. And again, if you have your textbook available on page 82 and 83, there's a great chart of what we're going to talk about. So the primary structure of the protein is its unique sequence of amino acids. So what is the number of links in the chain and what type are the links in the chain? That's what we call the primary structure. What is that sequence of amino acids that makes up the chain? And we'll look at each one of these individually too. The secondary structure, which you're going to find in most proteins, is along that chain, how is it kinked and folded and bent? That's what we call the secondary structure. Where are the coils and folds within that chain? Tertiary structure is determined by the interactions among the side chains. So remember, we have these little side groups, R groups, that are sticking off of the main part of the chain. Well, once they start interacting, they affect how the protein is bent and shaped, and that's called tertiary structure. And then finally, if we have more than one polypeptide chain that's all combining together, we get what we call quaternary structure. And quaternary structure is when you have multiple chains and how they interact as they are bent and folded. That creates your final level of structure. So we'll look at each one of these just really briefly. Here's a quick animation about protein structure. You would right-click and hit play. Let's go to the primary structure. The primary structure is basically what is the chain made of? What are the different amino acids within that chain? And what is the order that they're put in and how many are there? That's what you call your primary structure. So here we have the primary structure of this particular protein. I don't even know if I can pronounce that. But that is basically a listing of what are all the amino acids, what order are they in, and how many are there before you get from the beginning to the end. That's what we call primary structure. The sequence of amino acids, kind of like the order of letters in a long word. If you're spelling the word dictionary, well, the fact that there's a D first, then an I, then a C, then a T, on down the line, that would kind of be like primary structure. This information is inherited. It's given from one cell to the next. It's genetic information. It's predetermined. So this is the number and type of links within that chain. And we have a short animation here. This is primary protein structure. Right-click and hit play. Now, secondary structure is how does that chain bend and fold and turn? Because they're not just long, straight line chains. The different coils and bends and folds are usually the result of a hydrogen bonding that occurs between certain parts of the chain. So it kind of folds back on itself and bends and twists, and that's called secondary structure. And there's usually two different kinds. One of them is a coil, like think about the shape of a screw or a corkscrew. A coil. That's called the alpha helix. And then the other one is a folded structure called the beta pleated sheet. And that's more of a flat sheet-looking structure with hills and valleys. We'll take a look at each one of those as well. So here's a quick video of secondary protein structure. Of course, just right-click and hit play. Here is a picture. At the top, we have the alpha helix secondary structure. And you can see how it forms like a corkscrew shape. And then down here at the bottom, we have what's called the beta pleated sheet structure. And you can see how different hydrogen bonding causes a different shape. It's kind of like you've taken a piece of paper and you've done one of those origami folds to make a fan where it has hills and valleys, but it's on a flat plane. That beta pleated sheet, let me show you here. This beta pleated sheet, think back when we talked about the difference between starch and cellulose and how that was either called an alpha or beta glycosidic linkage. Well, that's very similar to what we see here. The alpha linkage, remember, was starch, and most animals and people can dissolve and digest that starch. But remember, the beta linkage produced cellulose, which humans and most other animals can't digest. The beta linkage tended to be quite a bit stronger. Well, we actually have the same thing going on here. If we have amino acids that are linked together and we have these polypeptide chains that have a secondary structure that is called beta pleated, it's very strong. So the proteins that are the silk of a spider's web, which are stronger than a piece of steel of the same weight, they're so strong because of this beta linkage, this beta pleated sheet structure. So that should kind of remind you, think back, look for these big ideas that kind of link between these various things. And so here is a pattern where this beta linkage lends itself to much greater strength. And so when we were talking about carbohydrates and the beta glycosidic linkage, that formed cellulose, which was a structural material. Now we're talking about proteins, a different type of macromolecule, but again, here we're talking about a beta linkage called pleated sheet, and that leads to structural proteins. So like the spider silk, which is very strong, similar to the cellulose was for the carbohydrates. The third level of structure is called tertiary, and tertiary involves those R groups. Remember, we have kind of the chain that stays the same, but then off of the main part of the chain we have these various R groups, a different R group for every amino acid, and there were 20 of them. So these R groups, they interact as well, and there's different types of bonding that goes on between these R groups, and that interaction causes even more folding, bending, shaping of the protein. And usually we have a wide variety of reactions going on here, or attractions, interactions going on here. We can have hydrogen bonds, we can have ionic bonds, we can have hydrophobic interactions, and we can even have what we call van der Waals interactions, which involve just those very slight and usually very brief positive and negative attractive and repellent forces. And then we can also have a fairly strong covalent bond called disulfide bridges, which may also affect these R groups and affect the shape and final outcome of this three-dimensional shape of the protein. So again, another quick animation. Hit right click and hit play to learn more about the tertiary structure of the protein. And then here we have a picture. So this looks just like the ribbon model, but we have some different folding and shaping, and the three-dimensional shape is affected by the tertiary structure, or the interaction between the various R groups. This picture is just another model kind of showing you some of the various interactions. Here we may have hydrophobic interactions, van der Waals interactions, hydrogen bonds, ionic bonds. There's a wide variety of different types of bonding that can go on between these little R groups. Remember, the R groups are the ones that are sticking off of the polypeptide chain, and so there's different interactions that can affect the shape of the final molecule. And then last but not least, we have quaternary structure. And quaternary structure only comes into play when you have two or more polypeptide chains that are clumping and forming together to form one massive macromolecule. A good example is collagen. Collagen is a fibrous protein. It's a structural protein, and it's made up of 3 polypeptides that are coiled together like a rope. I think I have a picture right here. So here's collagen. Here's a model, and there's an actual microscope photograph. Collagen is like the connective tissue within your body. It's actually probably one of the most common proteins within your body. It's about 40% of the protein in the human body, and it makes up the connective tissue of the skin, the ligaments, the tendons. It's very strong and very tough material, and it's basically, like we said, it's a chain of 3 different polypeptides linked together, and because there's more than one, it would have quaternary structure. Another good example is hemoglobin. Hemoglobin is the protein that we already mentioned. It's a transport protein, a globular. So globular means kind of spherical or blob shape protein consisting of 4 polypeptides. 2 of them have the alpha, and 2 of them have the beta chains, alpha linkage and beta linkage. So 2 of them would be kind of that corkscrew shape, that would be the alpha, and then 2 of them would kind of be that pleated sheet beta linkage or beta chain. I think we have a model here. So of course these are red blood cells down here on the right. So this is obviously not to scale because the red blood cells would be far larger than the hemoglobin itself. But inside those red blood cells are this hemoglobin protein. And it is a combination of 4 different polypeptides. And it's containing the iron that you see. And this is the protein, the molecule that allows for transportation of oxygen throughout the body. A quick little animation. You can right click and hit play. And then let's look at what would happen if we change that 3D shape. So you may have heard before of sickle cell anemia or sickle cell disease, and it's a change in the protein of the blood. So a change in the primary structure, and that affects what the protein can do and how it's able to function. It's an inherited disease, so if you have parents or a parent that has sickle cell disease, it is inherited. So it's not something that you could catch from someone else. It's passed on via your genetics. And it results from a single amino acid change or substitution in the hemoglobin protein. So there's a huge long chain of amino acids that determine this hemoglobin protein. One single link difference leads to this disease. So here we have an excellent kind of diagram that shows you. So primary structure, remember, is what are the links and how many are there? Remember, we have 20 different amino acids to choose from. So in normal hemoglobin, here is the primary structure. And then in the sickle cell hemoglobin, look, the only difference is right here at the number 6 spot. There's a single amino acid difference. In other words, a single link in the chain is different. Well, that has a major effect. The secondary and tertiary structure, which is like the 3D shape that it takes, that gets changed. And then the quaternary structure. Remember, we have 4 different chains all combined together. Well, that gets changed as well because these first 2 are changed. And then that ends up affecting the function of the molecule. If we have regular hemoglobin, the molecules don't bunch up, they don't affect each other, they each carry their own little oxygen and they all do their own job and they're happy, happy, happy. And a red blood cell with normal hemoglobin looks like this up here on the upper right. If you have sickle cell anemia, that affects the shape of the final protein, and that affects what the actual molecule looks like. The molecules form these fibers inside, and they have an odd shape. They have this sickle or sort of C shape to them, and that causes problems because they tend to bunch up together and form crystals, and they tend to not be able to carry as much oxygen, and they actually tend to get caught in the capillaries of the body as well. So if you have sickle cell anemia, you generally have some problems because your body is not able to carry all the oxygen that it needs as well as it should. I know it seems pretty impossible that one link difference in a very long chain of a 4-chain protein could cause such a problem, but it does. So it goes back to explaining how the primary structure, the makeup of the chain as far as what are its subunits, what are its links, what are its amino acids, and their order will affect how that chain is folded and shaped, which that will affect its different structure, and then that will affect how it works or if it can work at all. So these red blood cells that have this particular shape, they can work, but not nearly as well, and there's a number of problems, and that all goes back to this one single change in the primary structure of the amino acid. Or I should say, the one difference in this one amino acid in the whole polypeptide chain. In addition to that primary structure, what are the links in the chain and what is their order and how many of them are there? In addition to that primary structure, there are other outside conditions that can affect the shape of the final protein molecule. And so there are outside chemical and physical conditions like pH and salt and temperature and environmental factors can cause that protein to unfold or unravel or to shape into a different form. And the loss of the protein's original shape, its native structure, is something that we call denaturation or denaturation. Denaturation or denatured proteins are non-working proteins. They're biologically inactive. They can't do their job because they don't have their proper shape. Sometimes that can be reversed, so we have denaturation and renaturation here in this big circle. Here you have the normal protein on the left, and it's folded and shaped in a certain way, and when it's denatured, it unfolds or becomes a different shape. This protein is the same polypeptide chain, but because it's not shaped properly, it will not work. It's not biologically active. Some good examples of this. If you look at the eggs, the white portion of the egg, which is kind of this clear gel-like material, when you add enough heat, that protein, which is the ovalbumin, which is a storage protein we talked about, that ovalbumin protein actually is denatured. It's changed. The shape of the protein is changed, and it goes from this kind of clear jelly material to this opaque white material that you see here. So that the heat from the oven or the heat from the stove changes the protein and changes its shape, makes it unravel, and it changes the way that it looks. Relate that to, say, getting sick and having a high fever. You may have wondered what causes or what's the problem with the body temperature rising above 98.6. Well, when it gets to a certain level, say when it gets to 104 or 105, that heat, that temperature can actually start to denature proteins in your blood. And so you have proteins within your body that began to lose their shape and fall apart, basically, and that denaturation of proteins within your body can lead to your death if your temperature goes up and stays up for too long. So one big question that scientists often have is if they know the protein's primary structure, in other words, if they know what are the amino acids in that chain and how are they put together, in what order, can they guess what the final 3D shape of the protein will be? And that's really hard to do. Knowing the length of the chain and the links in the chain doesn't easily tell you what all is going to happen once the folding and twisting begins and what is the final shape of the protein. So most proteins actually go through probably a couple different stages before they get to this final stable three-dimensional structure. And there's a type of material, a protein molecule within living creatures called chaperonins. And that word may sound familiar, like from the word chaperone. Chaperonins are certain protein molecules that actually help with the whole folding process of other proteins. And there are certain diseases like Alzheimer's and Parkinson's disease, and you maybe have even heard of mad cow disease. These are all associated with proteins that are not folded properly. Remember, when a protein isn't in the right shape, when it's not folded properly, it cannot function the way it's supposed to. And so some of these diseases that we just mentioned are associated with misfolded proteins. And the body uses chaperonins to help make sure that proteins are folded in the proper manner. So similar to the word chaperone, chaperonins kind of provide a safe environment for these proteins to fold, kind of keeps bad influences away from them. And the way it works is these chaperonins sometimes actually form like a small container. So here we have a chaperonin in an E. coli bacterium cell, and this chaperonin kind of forms a container. The polypeptide chain is put into this container and sort of capped off from the rest of the environment. And then within that capped off portion, the protein can fold basically without any sort of negative interactions with the outside world. And when that protein is folded into the proper shape, the chaperonin basically comes apart or opens and releases that protein. So a great question that students often ask me is, well, how do we know this? I mean, we're talking about molecules. Even though they're macromolecules, they're so small, they're almost impossible to see, even with some of our best microscopes. How do we know that these proteins have certain three-dimensional shapes, and what are those three-dimensional shapes? Well, there's a couple of different ways that scientists use to look at these shapes, and there's no way you can just look through a school microscope and see these shapes, but we use a process called X-ray crystallography. X-ray crystallography is a very common one where we take the protein and we crystallize it, and then we use an X-ray machine to bounce X-rays off of it, and we get an image that we can kind of dissect and digest to determine the protein shape. Another way we can do that is with what's called nuclear magnetic resonance spectroscopy, and that's another way that we can look at these proteins' shape. And then a third way is to simply use computer power and to try to determine if we know the primary structure of the protein, can we use our knowledge and the computer knowledge to kind of decide what will the final structure be. And to do that, that takes an enormous amount of processing power. But we now have computers that do bioinformatics that can, to some degree, determine what the final shape of the protein will be just based on what is the primary structure of that protein. We'll end with a quick picture here of finding out, say for the example, the shape of this particular protein molecule, an enzyme that's called RNA polymerase, which is very important. We're going to find out to the genetics of your cells. So what they did is they crystallized this particular protein. They shoot X-rays through it, and then they have a digital detector that gives them this X-ray diffraction pattern. Now, obviously, this does not look like this 3D model that you see that they've created down here. But using this information, and I will be completely honest and say with information and knowledge that is above my head, they're able to discern what the three-dimensional shape of that protein is using this X-ray diffraction pattern. We're going to stop right there. I know proteins is a big, heavy topic with lots of information to digest. So take a break, come back, and we'll do the very last one, nucleic acids, in just a few minutes.